Hi everyone, my name is Jocelyn, and for this project I chose Salmonella enterica as my topic. The title of my article is Impact of Interventions on the Survival of Salmonella enterica in Pork. Listed below are the authors and where the research took place. Here on the first page, the authors talk about the use of Salmonella enterica serovar isolates, which were compared to other Salmonella serovars and non-pathogenic E. coli surrogates. These were compared in order to test their resistance to commonly used pork carcass interventions, their survival in ground pork, and their thermal resistance in ground pork. Salmonella enterica became an important point of research to the pork industry after a foodborne illness outbreak in the northwest U.S. that was connected to roaster pigs. All of the pigs came from the same establishment in Washington state and in 2015 caused 134 cases of salmonella in humans. While the facility was following industry standards, they were not taking precautions in regards to microbial interventions. The investigation showed that chefs in charge of cooking the pork had indeed ensured that the meat was brought to the correct temperature, but this only in a roundabout way led them to the conclusion that the strands tested were not any more resistant than the ones previously observed. The Iowa State University Veterinary Diagnostic Laboratory noticed that the higher prevalence of Salmonella enterica suggested that it was present on swine farms, thus it was already in the meat when it was eventually processed. Here's a little bit of info on roaster pigs. Roaster pigs are typically young and about 30 pounds to 60 pounds. They're usually roasted whole on a spit over a flame, like the picture on the right, or in a large roasting box, like a picture on the left. Here we can see that Salmonella enterica is rod-shaped and has very long visible flagella along the exterior. The problem in my chosen article was that people were falling ill from pork that was processed in the Washington state establishment. The authors wanted to see if one mixed culture of Salmonella enterica serovar isolates was more resistant than another mixed culture of Salmonella referens and E. coli surrogates. I believe that the hypothesis and reason for study was the importance of not letting Salmonella from these pigs spread more than it already had. It was important because although the meat was being cooked thoroughly, the bacteria was still spreading as evident by the 134 human cases. What is known so far about this issue? We know where it came from and how it was spreading, the Washington state facility, and through the larage of abattoirs that housed different animals from multiple places over long periods of time. We know the meat was cooked and heated thoroughly to an optimum temperature that should have killed the bacteria. We know that there were no differences in the responses that were observed between both groups of Salmonella serovars and the non-pathogenic E. coli surrogates with the intervention types, and that there were no differences in the recovery, survival, or heat resistance that were observed between both groups of serovars and the pork that was ground and stored at 5 degrees Celsius for 2 weeks. And we also know that the heat resistance of both groups of Salmonella serovars significantly decreased after being stored. One part of the method used in this study is the intervention treatments. Inoculated pork was treated in 6 different ways, not including the control, which had no treatment at all. There was a tap water rinse, a hot water rinse, a tap water plus 2% acetic acid, tap water plus 2% lactic acid, hot water plus 2% acetic acid, and hot water plus 2% lactic acid. Each of these samples was placed into a centrifuge and vortexed for 10 seconds. After being removed, they were rinsed with a 0.1% buffered peptone water in order to neutralize any residual acids. The first column on the left is the list of interventions that were used. The second, third, and fourth columns, respectively, are the reference Salmonella samples, the Salmonella samples from the establishment, and the E. coli surrogate samples after each of the interventions. There were no differences in population reductions that were observed between the 3 bacterial groups, but there were differences in population reductions found between each individual treatment. The tap water rinse was the least effective form of intervention used to reduce population. The hot water rinse that was followed by either organic acid, that being acetic acid or lactic acid, ended up being the most effective for reducing populations of all bacteria groups that were tested. This was shown to be consistent with the results from previous carcass intervention tests in both pork and beef. These studies have proven that there is no evidence to suggest that the Salmonella samples from the establishment are any more resistant to commonly used interventions in the pork industry. However, the reduced heat sensitivity of the Salmonella cerevars after cold storage could have more implications in the meat industry in the future.